Swayam Prabha. Digital India. Educated India.
Back. Shall we continue? So we were talking about genome editing, right? So we were talking about the chemistry behind that, right? So we were trying to understand like what is the chemical reaction that happens there, and like both in terms of the actual chemical reactions and the moieties that perform, like enzymes. But now we'll move into the actual genome editing, right? And a little bit of biology behind that. So as I said, I am going to represent my DNA with this railway track, right? Now, this genome editing is not something new. When I asked, does somebody know genome editing? I think one of you responded, there are some recombinant DNA technology. So, it is not something that is very new. It has been there since like 70s, where people have been using some technologies to kind of edit your DNA, right? So, if you look at all the, you all looked at GM crops. You know what are GM crops? Genetically modified crops, right? For example, the tomato that you get these days is not the actual tomato. It has been modified many times, and that you have a like red, pulpy, nice tomato. If you have seen it like 30 years back, it would have been a green, lightish green, or in the sense not green, but it will not be like as red as now, right? And there have been a lot of cotton variants. All this there have been genetically modified. Genetically modified means they are modified at the DNA level to improve their efficiency, to improve their productivity, to become resistant to a lot of these pests and other kind of things, right? So this is known for a long thing. But the general way that these do is, now I have told you two nucleases, endonuclease and exonuclease. Which one will be more useful? Which one should we talk about, endonuclease or exonuclease? Endo, because exonuclease does only at the termini, right? So that means you can only modify the bases at the end. But we know a lot of things actually, each of the gene would give rise to a protein. So if you want to modify a protein, suppose you probably have to modify somewhere in the middle. So endonucleases are the enzymes that we are going to talk about, because these are the enzymes that will modify the DNA in the middle of your DNA segment. It doesn't have to have a free OH to do their activity. And these endonucleases have been known for a long time. As I said, there was already a Nobel Prize for endonucleases discovered in 1978. A lot of these bacterial enzymes, right? A lot of the bacteria have these enzymes, and there was a Nobel Prize long back for discovering these endonucleases. So there are two, but the endonuclease, as I said, endonucleases are enzymes, right? They are very, very specific to where they actually bind to. Now, if you want to kind of this is your entire DNA sequence, and this is the red one that you want to modify, or you want to break, or you want to kind of do a phosphodiester bond hydrolysis, right? So there are endonucleases which kind of bind anywhere and then they diffuse to that spot and do the hydrolysis reaction. Or there are kind of specific DNA that exactly bind to this and then do the hydrolysis reaction. That means for hydrolyzing every basis, or for hydrolyzing a specific region of bases, you need to have different endonucleases. That means, for example, this is a list of endonucleases, a small list of endonucleases. There is an enzyme called BAMH1. It only hydrolyzes the diester bond if the DNA sequence is CGA-TTC, right? You all know what I am talking about, right? CGA-TTC is the alphabets, that is the code of your DNA, right? We talked about in the beginning, C, cytosine, right? All those things. There is other, suppose there is something called eCorV. It only does this, right? So that means for a specific region of DNA to be modified, you need to have a specific endonuclease or enzyme. That means suppose you want to have I said there are 3 billion bases in human body, right? Now I want to modify modify a certain part of it. That means I need to have so many, or even a fraction of so many enzymes, because an enzyme is very specific to where it is binding, right? So it cannot, that is impossible that you will have so many enzymes to do a restriction, or this is called restriction. This is called restriction digestion, okay? To do the chemical reaction on this. There are again specific enzymes where here the enzyme is binding to a specific segment, but it is also doing a hydrolysis at that segment. But there are enzymes which bind a slightly far away, but so where they do the hydrolysis reaction is immaterial, but where the bind is very, very specific. So there are also very specific ones. And then there are again something called non-specific. Now there are non-specific means it is like doing a chemical reaction in your test tube. You have your DNA, add your enzyme, it will cleave all the phosphodiester bonds possible. So both of them are not useful, because if it is very, very specific, the problem is for every DNA sequence that you want to modify. As I said, you all know human genome has around 3 billion bases. That means if you want to modify a specific one, you should first look whether there is an enzyme that recognizes that part or not. Right? And suppose somebody wants to modify some other region, you have to go and see whether that particular enzyme is there or not. And these are obtained from, we don't have this much. These are all obtained from bacterial species. So there are only limited number of such enzymes, right? So it is almost impossible that there will be, if there are n base pairs, there will be n or a fraction of n number of enzymes to do that, right? That is not going to work out. Yes.
Sir, what is N in the nucleotide?
N is any nucleotide. So when I write a specific one, GGA, that means that specific nucleotide. N can be any nucleotide, A, T, G, C. It could be any of those. That means it needs GCC to bind, but whatever is there after that is immaterial. It will do the hydrolysis reaction.
So one more thing is, what is a codon?
Codon is basically, now if we go back to this, DNA goes to RNA, RNA goes to protein, right? So 3 bases of your RNA. So our ribosome is the enzyme that kind of translates your nucleic acid sequence into amino acids. So for that reaction to happen, so the ribosome recognizes 3 bases at a time. For every 3 bases, it converts into a specific amino acid. So that 3 bases are called as codons. For example, here you see, if there are A, that will be converted into lysine amino acid. If it is G, it is converted into basically glycine amino acid. Okay, so these are called codons, where a ribosome recognizes 3 bases at a time and converts those 3 bases into a specific amino acids. In the sense, that is why I call this is the information. So the protein that you want, the information is here in the DNA. So DNA converts into RNA. What is based on the sequence of your RNA, that will give you the protein, because 3 bases specifically give a specific amino acid. It is not a random combination. Okay, that is called codon. Okay, now coming back to this. Okay, so there are 2 things. Yes.
So, for example, in DNA, since we have AT-CG pairing, can we reverse the pairing? Why, if like supposing we have CG, can we pair it as GC as well in the next
yes,
one? Or
yes.
Is it,
so G can pair with C, C can pair with G.
so either way in the same
Yeah,
DNA
if you look at the pairing is basically because again there is a hydrogen bonding network. G and C forms 3 hydrogen bonds between them. Whether it is C this side or C this side doesn't matter. C and G will form the pairing. Similarly, A and T will form the hydrogen bonding. So it doesn't matter AT-TA or GCCA, but A will not hydrogen bond with C. There will be, but it will be very, very weak that it is not going to be, it's a transient interaction.
Okay, so
Yes.
So could G and G bond with each other?
G and G cannot bond with each other. No.
So then why is there in the sequence 3G and then CC at this side?
Which side?
The first sequence.
Huh.
3G, AT, CC.
No, this pair, okay, so GGA, TCC. So the one that is horizontal is basically this sequence. That means guanine, cytosine, thymine, right? So if I go back to my DNA sequence, okay, so GGATC, this is this sequence which is related by the phosphodiester bond. But what is in the below? Like, so if I have written like this, right? So I am writing it as GGATCC. Now these are linked by the phosphodiester bonds. That means this is G, G, A, T, C, C. Now what will this hydrogen bond to will be C, C, T, A, G, G. So this vertical relation, vertical bonding is the hydrogen bonding. The horizontal one is the covalent phosphodiester bond. This can be anything because it is a phosphodiester covalent link. This is only hydrogen bonding, that is specific. Okay. Yes.
One more doubt. So why is it that the uracil is found only in RNA and not in DNA?
Okay, those are a lot of reasons for this are evolutionary in nature. In the sense, if you see the difference between thiamine, so A, T, G, C are in DNA. So A is there in that, G is there, C is there. Only T is not there. T becomes U. And if you see the difference between T and U, thymine and uracil, there is only a methyl bond difference, right? Where is my, So if you see the thymine and uracil, AU, this is thymine, and then you will have a uracil which will have this is the group, right? Additional, the methyl group is not there, here the methyl group is there. That is only difference. That actually become more, in biology, some of the things are evolutionary in the nature, but this is also one of those examples. Right? Chemically also you could see that when there are extra methyl groups or something, they'll start repelling each other. So you don't want to have an additional steric hindrance. Already there is a steric hindrance from the OH groups. Okay, now coming back to this story. So we have looked at two different. One is where each enzyme will recognize a specific DNA sequence and then it will cleave that. The other one is non-specific, which will break all the phosphodiester bonds. Both of them are not useful for us because one is you will have to have n, if there are n base pairs, you need to have at least a fraction of n, that means n by 3 or n by 6 number of enzymes. Those are quite a lot of enzymes. That means for every base pair you want to edit, you have to have a different enzyme. That is not feasible. And then if it is non-specific, then it is going to degrade everything. That is also not useful. So basically all these enzymes, these are specific endonucleases where they will bind to a specific region and then do the hydrolysis. But if we can develop something which is a non-specific endonuclease, but it will not go to all the phosphodiester bonds and cleave, you direct it where to go, right? Then basically if you want to edit a specific DNA sequence, you direct it where to go and then there will be a non-specific endonuclease. So that is what this CRISPR technique is all about, trying to guide the molecules to a specific region where the molecule will, the endonuclease will go to that specific region and do the hydrolysis. Is this part clear? So this is your normal endonucleases which are very specific to a very specific region. But if you want to have if you want to edit the entire genome, right, at different parts, you have to have the governing capacity. In the sense, you should tell where this endonuclease should go, and that you are telling by using something called guide molecule, which we will talk about. Okay, so this is what your CRISPR-Cas9 will do about. Now, before going into CRISPR-Cas9, so I want to tell you the story of how this was discovered, because in your textbooks, very rarely you see how things are discovered. You only see what they do, what is the scientific philosophy between that and how they are been. But you should also understand that most of the discoveries are serendipitous in the nature, in the sense like you do something and then it has been applied to a very different reason. So the CRISPR-Cas9 system is about understanding this specific molecular system which guides or takes it to a region and does the endonuclease. But let us see how this CRISPR-Cas9 system has been discovered. Okay. So just to tell you again some numbers. So the CRISPR-Cas9 discovery, I told you 1979 endonuclease got a Nobel Prize, right? CRISPR-Cas9 got a Nobel Prize in 2020 for chemistry. So all the so this was like 41 years, but it has been it got a Nobel Prize in 2020. I guess all of you know what is a Nobel Prize, right? It is the pinnacle of your scientific establishment. In the sense, if something is recognized by Nobel Committee, that means it has a high value in terms of its science. Okay, so this, and also before we go into it, So we should also see that the amount of time it took. So basically, this entire work on CRISPR-Cas9 started in 2011, right? So it started in 2011 and it got a Nobel Prize in 2020. So less than a decade of work. And if you look at the history of Nobel Prizes, generally the amount of work and the work that needs to go into Nobel Prize takes at least 30-40 years. That means 3-4 decades. But this is one of the rarest incidences where the discovery of CRISPR-Cas9 system and its application and then directly to the Nobel Prize took less than a decade. This shows how important it has, like in the sense it has actually changed a lot in terms of our scientific understanding. Okay, so and also that as I said, this particular Nobel Prize has a lot of interesting things. One is less than a decade, and this is the first time that 2 women scientists got a Nobel Prize. Generally, it is one male, one female, or 2 men. But this is something that is very interesting in the sense it never happened in the history of Nobel Prize chemistry that 2 women scientists got the Nobel Prize. So this is also something that you should keep in mind. In the sense, like this is a really exciting work that led to this entire story of CRISPR-Cas9. It has its own beauty in that, and so this is the story of CRISPR-Cas9. Okay, let us see. As I said, this CRISPR-Cas9 story is all about having a molecule that can guide you to any specific region of DNA, and a non-specific endonuclease will do the hydrolysis, right? Now, how was this even discovered? So the story began much before, sometime in 87. Okay, so when these 2 gentlemen, Francisco Mojica and this guy called Yoshirumi Ishono, so these are what they observed is that bacteria, so that is the observation is very important, right? So they have started, so you know, we can do our genome sequencing now, right? So you basically, you can get your DNA out and you can sequence your entire DNA and you can predict what kind of diseases you are prone to, like what is your ancestry, right? All those things you can do, right? So, but this is much before in 1980s where they could look into the small fragments of DNA only. So they looked at this as a very peculiar form of DNA. So what they looked in bacteria, not all bacteria, but many bacteria, they found that there is this DNA where a long DNA fragment where there are some palindromic repeats. You all know what are palindromes, right? What are palindromes? You read from the forward or the backward, they read the same, like madam and all those kind of words, right? So they observe that there are these palindromic repeats, and then there are some DNA. So again, whatever railway track I am showing is a DNA base pairing, okay, double stranded DNA. Okay, each of this vertical line shows that there is a hydrogen bonding between 2 bases. Okay, so you have a DNA fragment and they observe that these black ones, which are palindromic repeats, so they are kind of interspersed between this some non-specific DNA. So this green, blue, this yellow, all these are some DNA, okay, but between this DNA there is this palindromic repeats. Now, if you are a scientist and you discover some gene like this, what will you concentrate on? Suppose you are doing an experiment and you found a DNA, right? And you found that there is a long DNA, there is some random DNA, then there is a very repetitive palindromic, then there is some random DNA, palindromic, random, palindromic, and this whatever is not repeating is not same. That means it is different. It is for understanding, I chose different colors. One is green, yellow. That means this sequence is not the same, but this sequence is exactly same. Now, if you happen to discover such DNA fragment, what will you focus on? You will focus on the palindromic sequence. That is what they did, right? They focused on this palindromic repeat, but they realized that this palindromic repeat is not going to have any influence, and they didn't understand why this palindromic, because it seems not to have any functional importance, or it doesn't relate to any of the functions of the bacteria as such. Yes.
Are the palindromes same or are they?
The palindromes are same. The palindromes are same, but whatever is in the middle is different, right? So as any scientist, we would concentrate on that, and they realized that this palindrome things is not of much use, in the sense they could not find what is actually the functional importance of that. But then much later, so what they realized is that this is where the name CRISPR comes. I'll talk about that. But they tried to understand the biological significance of this palindromic repeats, and they could not find any. So this is called as CRISPR DNA. They call it CRISPR DNA. Why is it called CRISPR DNA? It is a short form of clustered, regularly interspaced palindromic repeats. CRISPR is a short form for clustered, regularly interspaced palindromic repeats. So basically clustered in the sense they are all in a single gene because they are all clustered together in a single gene, regularly interspaced. You see, there is some spacing, so that is why they are called like regularly interspaced short palindromic repeats. So still the name is concentrating on the palindromic repeat. They are saying there are some palindromic repeats which are separated by some short regularly interspaced, so there are, which have some small spaces and all of them are together. That is where the name CRISPR came from. So this is what the CRISPR is. But quite a long time they realized that this is not actually having any functional significance, this palindromic repeats. But what they observed is that this is a specific gene, but there are genes that are corresponding before this palindromic repeats that have nuclease activity and helicase activity. So as I said, every gene codes to a specific protein or enzyme, right? What they realized is that when there is this particular sequence, this is palindromes, this they don't understand what it is, but there is a sequence before this which is basically coding for, we just talked, right? Coding in the sense, 3 DNA codes for an amino acid, right? So when you look at the entire DNA sequence, that is coding for enzymes that are acting as a nucleases and helicases. Now what are nucleases and helicases? Nucleases means enzymes that hydrolyze the phosphodiester bond, right? Nucleic acid. What are helicases? So DNA helicases are basically, you see the DNA is a double helix in nature. That means one strand, one strand. If it is like, if you want to look at the telephone wire, right? It's kind of helical. Now helicases will unwind it. That means they kind of, again these are bound by the hydrogen bonds only. So they are kind of, helicases are the enzymes that kind of break the double helix structure. They make it into a single helix. They kind of open up. So those are called helicases. So in biology, always you should remember is when a specific gene is, so this is all called like, this is like, to start with I said genome, chromosome, gene, DNA, right? So this entire thing is a part of a gene, right? Whenever a specific genes are associated, like for example, nucleases and helicases are coding for this, that means they have a function in the sense that if some protein coding region is very close to this DNA sequence, that means that proteins have some functional significance to that. So this means they have kind of started hypothesizing that probably this particular region has something to do with nuclease activity and helicase activity, because why will an enzyme like nuclease and helicase be so close to this? So they started thinking in that way. Yes. The part which is away from this palindrome. I said there is a specific palindrome and then there is an interspersed region, but there are also some beyond this palindrome which are not relevant to this, right? Which are not bacterial part, but those are coded, which are bacterial part, which are coding to helicase and nucleus. So it is further. Okay. Yes.
It will always be towards the end of
towards, yes, very close to it. So it need not be always at the end, it is close to this this region, CRISPR DNA region. Okay. So then basically what they realized is that, as I said, this is all part of this gene, Cas gene, as I said, Cas gene, which codes for nuclease activity and helicase activity. Then they start, okay, if palindrome is not there and there are these enzymes, nucleases and helicases, then it must do something with the breaking of this nucleic acid bonds and also doing the helicase activity of this, right? So then they realized, upon further experiments, they realized that whatever this sequence, the non-palindromic part, that means the spacer part, that is having a huge or very high correlation, or it is similar to viral DNA. That is what they have figured out. So to start with, they have focused on the palindrome. Just like anybody, if something is repeating, we will focus on the repeating part. They have focused on the repeating part and they realize that it is not of much importance. But they realize that there is some genes that are close to it which are doing the helicase activity and DNase activity. But upon further experiments, they realized that The short one is not some random DNA, it is a DNA of virus. Like that means if you take the viral genome, these small parts are actually from the viral. So basically this is a part of a DNA, so this is a part of a bacterial defense mechanism. How does it do that? What it does is, suppose this is a bacteria, right? Now a virus comes and it kind of introduces its DNA. In this case, it is basically it is introducing its, it can be either be RNA virus or DNA virus, but it is introducing this DNA. You know, you all remember, you all know that immunity, so suppose you have been, you get a infection of a specific virus one time, then you generally develop an immunity against that particular virus again, right? There are exceptions to that, but most of the times once you get a specific, for example, measles, chickenpox or these kind of things, once you get it, you are not going to get it again because your body has a memory to develop antibodies against that, right? So, same thing bacteria also has. So what it does is, so once the virus comes and then it kind of introduces its DNA. So suppose this bacteria survives that, what it does is, basically that will introduce the viral DNA into the bacterial DNA. So that means the virus came, if the bacteria survives, that has to be there. If the bacteria survives, the viral particles will leave some of its DNA into it. So it will introduce this DNA into the CRISPR DNA as I showed you. So that means it is a kind of, this is kind of a bacterial memory. That means, okay, this particular gene, oh yeah, this particular virus has come before. I know how to attack this, right? So how does it have a memory? It has a memory, it will store the memory in terms of DNA. So the bacteria, if a virus attacks the bacteria and leaves its DNA, that DNA is incorporated into this. That means it will be stored as a memory. If suppose the same virus comes again, the viral DNA is already with it, so it knows how to handle that, right?
So is that why before that there is nuclease and helicase activity?
Exactly.
It will go attack the...
Exactly, very nice. So basically that is what, so basically you have a nuclease and helicase, we are coming to that, right? So once it knows that and it is close to the virus, it will start going there and then cut the viral DNA so it doesn't propagate. So it's kind of a defense mechanism. So it realized that, so basically it will do this. And then how does it? So now this is called as adaptation. In the sense, bacteria has adapted to the virus. Now if the second time the same, but it doesn't leave it there. So as I said, when you want to use something as a defense mechanism, when you have guns, right? Like you want to have a defense mechanism. You know, similar like army people, defense people, right? You don't have to fight a war every day. So it doesn't mean that they'll keep their guns at the room and then whenever war comes, I'll go in. Then the entire gun would have been rusted and it will not even fire, right? So even if you speak of Navy people, even if there is a war or no war, they generally go with their submarines into the water and do a kind of a, they basically do a mock drill kind of thing, right? Just to make sure that it is happening. So because you don't wait for the real enemy to come to take your guns. And because by that time you don't know what is the function of that, right? DNA also, bacteria also does very similar to that. So it wants to keep its, this is a defense mechanism, right? So it wants to keep its immunities or it wants to keep its weapons ready. And as you remember, DNA is not a great weapon because it is a double stranded, it is tightly bound, right? DNA doesn't react. DNA is a highly unreactive molecule because it is a tightly bound, you know. The structure is a double helix structure. For unwinding that, you need a special enzyme. So by the time a virus comes and does all these things happen, it will take a long time, right? So when you're storing a weapon, when you want to store as a weapon, you want to store in the form where it is ready to attack. So the DNA transcribes this, or the bacteria transcribes this DNA into RNA, and then you see I wrote it as CRISPR DNA, but it has transcribed into RNA. So it transcribes and keeps it ready. So if suppose the next time the virus comes, it is going to attack it. So it keeps as a RNA and keeps it ready. So and then what it does is it will cut them into different parts. So for example, this particular DNA or RNA fragment, which is from this DNA, came from a different virus. This came from a different virus. This came from a different virus. So you don't want to store everything together. So it will basically cut that and it will store as a multiple weapons. Right? Weapons in this, I'll show you here. The weapon is basically a nuclease because if you can break the nucleic acids, then the RNA is no longer functional. So that is what I mean by weapon here. Right? So it will cleave that this information is from a specific virus. This information is from a specific virus. This is from a different virus. So it cleaves into different parts and then it stores in the form of basically RNAs, but RNAs themselves are not stable. They are highly reactive, I said. So it should be like you want something to be reactive, but not highly reactive, because only when the enemy comes, you want that to attack. But it should not be very stable like DNA. So it stores as RNA bound to a protein complex. This is called as a molecular scissor. This protein, whatever is coming from the, you remember there are specific genes that were coding for nuclease and helicase I mentioned. This is, whatever the structure I have shown you or whatever the cartoon I showed you is a cartoon of a enzyme which has both nuclease activity and helicase activity. That means this virus will store the RNA within the two enzymes, the helicases and DNases, helicases and nucleases, which are DNases, and it is ready to attack. Right? It stores in that so that RNA is not very reactive, but it is also reactive in case something comes, and you also have your weapons. In the sense, now next time when a virus attacks, right? So that was all adaptation. Now when's the next time a virus attacks? So this, because the greatest, again going back to the chemistry part, right? So there is A recognizes to T, G recognizes to C, right? There is a hydrogen bonding there. Now, if suppose this particular thing is basically there, and if the same virus comes again, this helicases will unwind, and because this is already previous, it knows that this particular sequence it already knows, so it will go and bind to that, and then the helicase will unwind to that, and then the nucleus will break it, and then it will basically destroy the new viral DNA. Okay, so then the virus is no longer active. So that is called as interference, and then this is called as. So this is basically how a specific class of bacteria fight against virus. Is this clear? Now, like we are talking about a prelog, right? So basically this is all coming from your viral part, where if a new virus enters, because it has already stored, this is again your ATGC kind of base pairing. It will go and base pair because RNA is more reactive. Once it base pairs, it kind of cleaves that, and then it does the nucleus activity and breaks this. This is all fine. Now, this is a defense mechanism. We also have a defense mechanism. Bacteria also has a defense mechanism. But what is it so interesting that it got a Nobel Prize, right? Like, why is it, what has it revolutionized? Every living organism has its own defense mechanism. This is one of the bacterial's defense mechanism against virus. Now, what is it so great in that it has led to the revolution, right? That is what we are going to talk about now. So that is basically the development of CRISPR-Cas9. So what I want to emphasize is the research that happened for the discovery of CRISPR-Cas9 has nothing to do with gene editing. It was actually a part of understanding the defense mechanism of a bacteria, right? But then people have used it. Can you correlate now? Like now you have discovered this. The original problem that we thought was we want some kind of enzymes where you can guide the nucleic acids to a specific region, and then they can do a nuclease and helicase activity. Now, how do you think this has been correlated to that?
I have a doubt. If the same virus, the same type of virus, mutates to have a different gene sequence and it attacks the same bacteria, would the CRISPR difference still work?
So it depends on what mutation. Mutations means conversion of one amino acid into other, or conversion of one base into other, right? If it is actually exactly mutating in the exactly same region as what it has stored, and if it is one base, suppose. So because there are 13 to 14 bases and each of them will hydrogen bond. If there is one base that is mismatched, it will still bind with slightly less efficiency. It will still act. Now, if there are like of the 13, if suppose there are like 7 or 8 bases mutated, then CRISPR cannot, because its defense mechanism works on the hydrogen bonding between them, right? So it cannot do anymore, right? But that has to be like very, very coincidental that it has to mutate only in the because virus doesn't know that this has been stored by the bacteria, right? So there is no way that it's a high chance, the chance of that happening are low, but it can happen where it only mutates in the region where it has stored, right? But it does happen, yes.
If the CRISPR is going to remove the viral DNA from the interspace sequence, how will the bacteria have the memory of that virus, and how will it again fight back and again the virus?
Sorry, say that again.
So enzyme is going to like remove the interspace sequence, and how will the bacteria have the memory of
The enzyme is, so you're talking about this step or this step?
the second step?
This one. So you understood that basically it stores everything in form of RNA, right? Now once a viral DNA comes or a viral RNA comes, this is kind of hanging, this is hanging, right? That means it is not bound to anything, it is ready to pair. RNA also pairs, right? It is ready to pair, but because of the repulsion it will not be very strong, right? It is ready to pair. So if the complementary sequence of this comes from the DNA of the viral DNA, now this is the new viral DNA, right? Now this, because this is the same thing that has been stored by this, so one part of it will be complementary to the other. Now it will recognize that complementary part and it will basically, this enzyme will cleave the bacteria of it. It is not going to cleave here, it is going to cleave somewhere else. And this part will, so this is for the defense next time when it comes. For the first time when it comes, it doesn't have this mechanism, right? It is just going to keep that and store it. When the next time it comes, because when the first time it comes, when adding is happening, okay, so when the adding is happening, you don't have the nuclease and helicase there, so it will just add to that. So the next time when it comes, it recognizes that and does that. Does that answer your question, or you want a little more?
So the nucleus
Cuts,
cleaves off
but
the-
it doesn't cleave off here, right? It cleaves off a few bases outside that.
So the memory is still held back with the bacteria, and that's how it helps.
But this part is not the part of the memory. The memory happened when the virus attacked last time, the first time. This is the defense. When the second time it attacked, this thing happened. So the memory is still there from the first time, right?
How is it stored?
In the form of your, this is how it is stored, in the form of RNA. Yes, right? This is only for defense mechanism for cleavage. So when the first time it enters, there is no, it doesn't have anything on that. So it will take a specific viral fragment and it will store it, that's all. But next time it comes, it does this nucleus activity.
Sir, how does the maturation take place?
How does the maturation take place? You mean this cleavage into small things?
Uh-huh.
Again, there are enzymes for that. So again, I didn't go into very details, but yes, there are enzymes that cleave off on the palindromic part. You need to cleave off at the, not in the interspacing part, you want to cleave it off at the palindromic part. So the enzymes that cleave off at the palindromic part. Okay. Yes.
So, if the viral DNA and the RNA is being stored inside the bacteria, would those segments still produce the viral protein? Would they still be active?
Oh, that's a good question. No, they will not be, because they are in the form of a complex with the protein. So only if it is a naked virus that is there, and if it is one of the RNA that the ribosome recognizes, it would. But in this case, it is only a small fragment. For producing a protein, you need the entire gene. Right? It is only a small fragment of the DNA. So it doesn't have, it is like a half the sentence, which doesn't make any sense, right? So you need to have the entire sentence to make a gene. So that part is not there, right? So it will not produce proteins. That number I cannot tell, but I think any bacteria can fight quite a lot. It does encounter a lot of viruses. So if you see a CRISPR bacterial gene, it is very, very long. In principle, probably few hundreds. I don't know about the number, but in principle, I don't see any limit why there. I don't see any reason why there should be a limit on it, because it is just adding to the already existing thing. Right? Okay, so now the question is, okay, this is all defense mechanism, fine. But how are you going to use this for the original question, in the sense, how are we going to use this to direct a specific, like, you know, the original question is, now you have a specific DNA. Now, if I have any specific endonucleases, I know where to cut. But I want to have a guide molecule which will guide to this, and there it cuts. So how do you think this defense mechanism would have given a thought? Like, this is a separate story, in the sense it didn't do anything with the CRISPR. The name is CRISPR, but it is a defense mechanism. It has nothing to do with the genome editing. How do you think they would have thought about this?
In this case, the helicase and nuclease activity is concentrated there because it already has a small sample of it plus the enzyme.
Yeah.
So if we are able to synthesize that sample and keep it ready, then
Very good. So that is how the entire discovery. So this was done in 1987 or 1990. People forgot about it. So these two ladies, Emmanuel Charpentier and Jennifer Doudna, people were working. They were working with various recombinant proteins. So they realized that, okay, why don't we use this? This is a bacterial defense mechanism. Why don't we use it for genome editing? At that time, genome editing was very, very in a high demand, in the sense like, as I told you, if you can edit a genome, if you can edit a gene, you can do wonders, in the sense you can improve the crop productivity. If you can edit a specific gene, you can make mosquitoes not like, the parasite will not go into the mosquitoes, or its reproduction will go down, or you could increase the crop productivity, or you could even basically if you want to specifically synthesize specific organs, you could use that. So all these are genome editing. I think from early 2000s and slightly mid 2000s, this has become a very, especially you might ask, before that, why was it not there? We all know genome is important, but 2000 is a major path-breaking year. In the sense, 2000 is the year where human genome sequence came into it. In the sense, people were able to sequence the entire genome. Previously, it was not possible to sequence 3 billion bases, but the technologies have improved so many times that if you tell me, I can get the entire sequence of my genome, right? So in early 2000s, there was a huge rush to sequence genomes. Now, okay, now you have sequenced genome, like what do I do with the data, right? That is what, I know, but can I, then the thought come, okay, I have the data, now I know which part of gene is giving which protein and which disease and other things. Now suppose I edit that, I can eradicate that, or I can improve something. That is that is how the thought process came, right? Now that is how this field came. In the sense, they took some old thing and then it is off on.